Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
In 1948, there was this medical paper that documented what looked, for all intents and purposes, like an absolute miracle.
Oh, I know the case you're talking about, the 9-year-old girl.
Exactly. So she was admitted to a hospital in a completely comatose state. She was suffering from what doctors at the time called malignant malaria.
Which is just a terrifying term.
It really is. I mean, she was delirious, unconscious, and her blood was absolutely teeming with the Plasmodium falciparum parasite. So the medical staff administered intramuscular and oral quinine.
And against some pretty formidable odds for that era, she woke up.
Right. And the medical record actually confidently stated that she, quote, went home in perfect health. And that single phrase, perfect health, it essentially helped cement a medical dogma that lasted for decades.
It's an incredibly compelling narrative. It plays perfectly into our desire for modern medicine to just be this definitive triumph over an ancient pathogen.
Just a clear-cut win.
Exactly. The story is simple. You cure the infection, you clear the parasite, and the patient goes back to their life exactly as they were before that fateful mosquito bite.
Right.
We've structured a lot of our global health metrics around this very idea, this idea of survival being the ultimate finish line.
But what if the true cost of that infection doesn't actually appear until, say, a full decade later?
That is the big question.
I mean, think about the last time you had a severe flu. You probably felt entirely cured a week or two later. Now, imagine if that flu secretly altered the way your brain processes complex logic.
And you wouldn't even know it.
Right. You wouldn't know it until 10 years from now. What if the toll of this disease is entirely invisible until that child, who seemingly recovered in perfect health, is sitting in a high school math class and finds herself fundamentally unable to process the equations on the board?
That invisible toll is shifting the entire paradigm of global infectious disease research today. We're finally moving beyond the immediate short-term horizon of survival.
Which is long overdue.
Completely. The medical community is looking at the long tail of infection now. They're recognizing that surviving a pathogen doesn't necessarily mean you've escaped it without some kind of structural biological compromise.
And the scale of this issue simply can't be overstated. I mean, we really need to look at the numbers to understand the gravity here.
The statistics are staggering.
They really are. In 2024 alone, there were 282 million reported cases of malaria globally, and that resulted in 610,000 deaths.
And the demographics of who that hits hardest is really important.
Right. The overwhelming majority of those cases, and about 95% of those deaths, occurred in Africa. And it predominantly strikes children under the age of 5.
Now, modern anti-malarial drugs like the artemisinin-based therapies have been revolutionary.
Absolute game changers.
For sure.
Yeah.
They've averted over 14 million deaths since the year 2000. But as we're about to unpack today, surviving the acute phase of the disease is merely the prologue to a much longer hidden biological story.
So for our deep dive today, we are unpacking a massive, groundbreaking longitudinal study that was recently published in JAMA, the Journal of the American Medical Association.
This is the MIND study, right?
Yes. The research team investigated the malarial impact on neurobehavioral development. The MIND study. We're going to explore the hidden toll of severe childhood malaria.
Specifically looking at how surviving these severe forms of the infection leaves a permanent imprint on a child's cognitive ability.
Right, and their academic performance 4 to 15 years after that initial illness.
To really understand how an acute fever in a toddler affects a teenager a decade later, we first have to look at the unprecedented way these researchers tracked the survivors.
Because longitudinal tracking in global health is notoriously difficult. I mean, people move, contact information changes, local infrastructure shifts over time.
Exactly. Finding 889 individuals a full decade after their initial hospitalization is a Herculean logistical feat.
It really is. So researchers in Uganda traced these children and adolescents who had previously been part of 2 older, distinct severe malaria studies.
And those took place between 2008 and 2018.
Right. So they brought them back and ran these incredibly comprehensive cognitive and academic assessments. The average follow-up time was 8.4 years.
And it's crucial to note here that they didn't treat malaria as just a single monolithic disease. Malaria presents on a really wide clinical spectrum.
So they broke it down into specific groups.
Right. The researchers stratified the survivors into very specific clinical phenotypes based on how sick they were during that initial hospitalization.
Okay, let's go through those.
First, they looked at children who survived cerebral malaria. This is the most critical form, characterized by impaired consciousness and coma.
That's the most severe presentation.
Exactly. Second, they looked at severe malarial anemia. This is where the parasite aggressively destroys red blood cells, driving the child's hemoglobin to just dangerously low levels.
And what was the third group?
Finally, they looked at a group they categorized as other severe forms. This included children who presented with respiratory distress, complicated seizures, or extreme prostration, but who importantly never entered a coma.
Now I have to pause here and introduce a major analytical challenge. Whenever researchers discuss long-term academic tracking, the environment plays a massive, undeniable role.
Oh, absolutely. The socioeconomic variables are huge.
Right. So if I'm a researcher testing the academic performance of teenagers in regions with highly varied educational infrastructure, how do I know their test scores are a result of a malaria infection they had a decade ago?
Right, instead of just the local school system.
Exactly. Couldn't those scores just be a reflection of their socioeconomic environment? I mean, factors like the quality of their local school, whether they have electricity at home to study at night, or their parents' income level, that could easily skew the data.
That is the Achilles heel of almost every observational study out there.
Because it's so hard to control for.
Yeah, right. If you compare these survivors to a generalized national average, the data is basically useless because the socioeconomic variables drown out the biological signals.
So how did the MIND study fix that?
The methodology addresses this brilliantly. The researchers utilized a highly specific control group comprised of what they called community children.
Community children, meaning what exactly?
These were healthy children recruited from the exact same neighborhoods and, in some cases, the extended households as the malaria survivors.
Wait, so they're comparing neighbors to neighbors?
In many cases, yes. By doing this, they effectively controlled for the socioeconomic environment. If the local school system was underfunded, well, both the survivor and the control child were dealing with it.
That makes total sense.
Yeah.
If the neighborhood lacked infrastructure, both children experienced it.
Exactly. Furthermore, their statistical models adjusted for age, sex, height for age, and weight for age ratios.
Oh.
And even the primary caregiver's level of education. They essentially isolated the severe malaria infection as the primary variable by ensuring the control group shared the exact same environmental baseline.
So knowing that the study successfully isolated the severe infection as the variable, let's look at exactly what happens to the long-term architecture of the brain.
The findings published in JAMA are pretty stark.
They really are. Children who survived either cerebral malaria or severe malarial anemia scored significantly lower in overall cognitive ability and math achievement compared to their healthy peers from the exact same community.
The data provides a very clear, quantifiable picture of this deficit. Across the comprehensive testing batteries, the researchers noted a cognitive gap amounting to a z-score difference of negative 0.22 to negative 0.49.
Okay, let's translate that for anyone who isn't living inside a statistics textbook.
Fair enough.
A z-score difference of negative 0.49. What does that actually look like in a practical, everyday context for these teenagers?
Right. If we convert those standard deviations into a more commonly understood metric, we're looking at a permanent drop of roughly 3 to 7 IQ points.
Which is a real tangible drop.
Exactly. Now, I know 3 to 7 points might not sound catastrophic if you're just looking at a single individual on a good day.
Right, you might not notice it in a casual conversation.
But on a population level and for an individual's long-term academic trajectory, it is a significant structural disadvantage.
It compounds over time.
It does. It can literally be the difference between mastering complex secondary school concepts like algebra and falling irrevocably behind in a highly competitive educational system.
Fascinatingly, this cognitive deficit wasn't just a uniform dulling of all mental faculties. It was really specific.
This is where the testing tools get really interesting.
Right. The researchers used validated tools, specifically the Wide Range Achievement Test, known as the WRAT-4, and the Test of Variables of Attention, the 2VA.
And what did those specific tests show?
These assessments showed that reading ability and attention spans were largely unaffected.
Which is so counterintuitive when you think about brain injury.
Right. The deficits were heavily concentrated in overall cognitive processing and mathematics. And even more surprising, the children who survived those other severe forms of malaria the ones who had respiratory distress or complicated seizures without entering a coma, they showed absolutely no long-term cognitive deficits compared to the community controls.
The specificity there is really the most revealing part of the entire study.
Why is that?
It tells us that severe systemic illness alone isn't what's causing the brain damage. If the sheer stress of a severe fever, respiratory distress, and hospitalization were enough to impair long-term cognition, that third group of children would have shown deficits as well.
But they didn't.
Exactly, but they didn't. The damage is directly tied to the specific pathophysiology of the first two categories: cerebral malaria and severe malarial anemia.
I'm trying to picture how this works functionally in the brain. It's almost as if the brain's foundational processing speed and logic centers the exact hardware required for sequential processing, holding multiple variables in working memory and executing mathematical operations. That specific hardware incurred permanent physical damage.
I like that analogy.
But the neural pathways responsible for reading comprehension and rote attention were somehow spared from the initial injury. Or maybe they were able to route around the damage and heal over time.
Like a computer where the RAM is permanently damaged, but the hard drive holding the vocabulary words is totally fine.
Exactly. The RAM is fried, but the storage is intact.
The hardware analogy is incredibly useful here, but we really need to look at what's actually breaking the hardware.
Right. The mechanism of injury.
If only specific forms of malaria cause this highly targeted cognitive damage, there has to be a unique biological mechanism happening during the acute infection.
The editorial accompanying this study, authored by infectious disease experts Drs. Sundarman and Odom John, really synthesizes exactly how this pathogen physically alters the vascular system.
Because it's not just a fever, it's a structural attack on the blood vessels.
Let's unpack that mechanism. How does a blood-borne parasite swimming around in the circulatory system alter the structural integrity of the brain's logic centers?
We have to look at the survival mechanism of the Plasmodium falciparum parasite itself.
Okay.
When it enters the human body, it survives by invading the host's red blood cells. It essentially hides inside them.
Indeed.
Very. But once inside, the parasite radically modifies the plasma membrane of that red blood cell. It produces specific proteins that push their way to the surface of the cell, making the cell physically sticky.
So instead of flowing smoothly like water through a pipe, the red blood cells turn into microscopic Velcro.
That's a great way to visualize it. In cerebral malaria, these infected, sticky red blood cells adhere to the endothelial lining. That's the inner walls of the brain's microvasculature.
So they're sticking to the walls of the tiniest blood vessels.
Exactly. They essentially clog the tiniest blood vessels in the brain. Think of the blood-brain barrier like a highly exclusive nightclub bouncer.
Okay, I'm with you.
Its entire job is to let nutrients in and keep toxins out. But these sticky malaria cells act like a massive mob that clogs the street outside the club.
And the bouncer gets overwhelmed.
Right. This sequestration triggers an aggressive inflammatory cascade. The resulting riot causes profound dysfunction of the blood-brain barrier, localized oxygen deprivation, and cerebral swelling.
So the bouncer's overwhelmed, the doors break down, and the brain tissue is damaged.
Precisely. And in severe malarial anemia, even as overall red blood cell production plummets and the patient becomes critically anemic, this same dangerous vascular adherence is still occurring throughout the body's vascular network.
It's just a systemic breakdown. The researchers actually identified specific biological red flags present at the time of the initial hospitalization.
Oh, the predictive markers. This part is fascinating.
It really is. These were acute clinical markers found in blood tests taken a decade ago that accurately predicted which children would struggle with math and cognitive processing today.
Like a biological crystal ball. What were the specific markers?
They identified acute kidney injury, hyperuricemia, which is an excess of uric acid in the blood, and elevated plasma angiopoietin 2.
It's quite a list.
And for children with cerebral malaria, elevated plasma tau was also a really strong predictor.
Elevated plasma tau makes perfect sense. It's a well-known direct indicator of neuronal destruction.
Right, because it's inside the brain cells.
Exactly. When brain cells are physically damaged or die, they burst open and release their internal contents, including tau proteins, into the cerebrospinal fluid and eventually the bloodstream.
So it's basically the debris of dead brain cells.
Yes. Seeing elevated tau at admission is a smoking gun. It directly correlates with the long-term cognitive deficits we're discussing because it proves brain cells were actively dying during the fever.
Okay, plasma tau makes complete sense to me. It's a brain injury marker predicting brain damage. But I am stuck on acute kidney injury.
It seems disconnected, right?
Completely. Why would a kidney failing during a toddler's fever predict that child's math score 10 years later? That sounds a bit like reading tea leaves.
I get that.
Help me connect those dots, because the kidneys and the brain are physically and functionally very far apart.
This is where we have to stop looking at organs in isolation. Severe malaria is not a localized event happening in one organ. It is a systemic vascular crisis.
It's happening everywhere all at once.
Exactly. Acute kidney injury, metabolic overload leading to high uric acid, and the release of angiopoietin-2 are all deeply interlinked pathways.
You mentioned angiopoietin-2 earlier.
Right. That's a critical piece of this puzzle. It's a protein that regulates the endothelium, which is the inner lining of the blood vessels.
Okay, so the pipes again?
Think of the endothelium like the non-stick Teflon coating on a frying pan. It keeps everything flowing smoothly.
Right, nothing sticks.
But when angiopoietin-2 levels spike, it's a marker that the Teflon coating is being ripped apart. It indicates severe endothelial damage and is a primary driver of decreased blood-brain barrier integrity.
So if the Teflon coating is damaged everywhere, the kidneys are just the first alarm bell to ring.
Exactly. The kidneys are essentially highly dense, highly vascularized filtration systems. They require immense, constant blood flow to function properly.
So they're super sensitive to vascular issues.
Highly sensitive. If the kidneys are failing due to microvascular blockages and endothelial inflammation, it's a clear, undeniable indicator that the systemic vascular network is under absolute siege.
I see.
If the vascular damage is severe enough to literally shut down the kidneys, it means the blood-brain barrier's defenses, that nightclub bouncer we talked about, have almost certainly already been breached.
The kidney injury is acting as a proxy marker. It shows doctors the severity of the invisible microvascular damage occurring inside the brain at that exact moment. It's incredible how interconnected the system is. But before we treat these biological markers as absolute diagnostic rules, we need to examine the scientific guardrails of the data.
Because no study is perfect.
Right. Every deep dive requires us to look honestly at the limitations of the research. First, this is a prospective observational study based in a single country, Uganda.
Which means it has certain constraints.
Because it's observational, it can't definitively prove a 100% causal link between the infection and the exact IQ drop, though the robust use of the community controls strongly suggests that causality.
It's a very strong correlation.
And second, tracking people over 15 years is incredibly messy. The study experienced a 25% attrition rate. They just couldn't find a quarter of the original participants.
We really have to consider the grim statistical reality behind that attrition rate.
What do you mean?
The researchers noted something crucial here. The patients who presented with the most severe disease during their initial hospitalization were statistically the most likely to have died before the 15-year follow-up could even take place.
Oh, wow.
This is a textbook, tragic example of survivor bias and longitudinal medical data.
Wait, meaning the data we're looking at, the 3 to 7-point IQ drop, that only represents the children whose initial infection was mild enough that they actually survived the next decade.
Sadly, yes.
That is heartbreaking.
It is. The children who suffered the most catastrophic, widespread neurological injuries from the malaria likely didn't survive to sit down and take the math and reading assessments 10 years later.
So the data is skewed.
Right. Therefore, the cognitive gap observed in the survivors might actually be a highly conservative underestimate of the disease's true, overarching cognitive toll on the population.
Because the worst cases aren't even in the math.
Exactly. If you factor in the severest cases that were lost to attrition, the reality of the neurological damage across all infected children is likely much, much steeper.
The medical community's perspective on this is shifting rapidly because of data just like this. One of the most revealing details in the study was regarding treatment protocols.
Right, the shift in drugs.
Over the 15-year span of the parent studies, the World Health Organization changed the primary treatment recommendation for severe malaria. They moved away from intravenous quinine and shifted to parenteral artemisinin.
Which is standard protocol now.
Right. The researchers analyzed the data to see if this superior modern treatment protected the brain better, and the result was stunning.
Let me guess, it didn't.
It didn't. Changing the primary treatment to a faster-acting drug did not change the long-term cognitive outcomes.
It's a deeply sobering finding, really. Artemisinin is incredibly effective at its primary job, which is clearing the parasite from the bloodstream and saving the patient's life in the acute phase.
But the brain damage still happens.
Right. The data tells us that by the time the child arrives at the hospital and the drug is administered, the microvascular damage, the adherence of those sticky cells to the blood-brain barrier, the inflammation, the tau release, it's already done.
The fuse has already been lit.
Exactly. Surviving the parasite is no longer enough if the brain hardware is already compromised.
So what's the next frontier then? If killing the parasite faster doesn't save the brain, what actually does?
The focus is shifting toward protecting the vascular system during the infection alongside clearing the parasite.
Okay, so a two-pronged approach.
Researchers are currently looking at manipulating endothelial angiopoietin signaling.
That's the Teflon coating protein again.
Remember that angiopoietin-2 protein that spikes when the blood vessels are under stress? The tactic being explored right now utilizes monoclonal antibodies.
Wait, isn't that an approach currently used in advanced cancer treatments?
Yes, it is.
Can you break down monoclonal antibodies for us? How does a cancer treatment apply to a mosquito-borne illness?
Think of a monoclonal antibody as a highly targeted, lab-made immune system soldier designed to bind to one specific target.
Like a heat-seeking missile.
In this context, researchers want to use these antibodies to target and neutralize the angiopoietin-2 spike.
To protect the Teflon.
Exactly. The theory is that if we can administer a treatment that chemically stabilizes the blood vessels, basically reinforcing that Teflon coating, we might be able to prevent the blood-brain barrier from breaking down in the first place.
So we provide chemical scaffolding to protect the brain's architecture, while the traditional antimalarial drugs go to work clearing the parasite.
That's the hope. It's a really innovative approach.
But with the biology and the data unpacked, what does all of this mean for public health? How does this change how we view severe illness on a macro level? Let's move into our myth versus fact segment for this deep dive.
I love this segment.
The prevailing myth, built on cases like that 1948 medical report of the little girl going home in perfect health, is that if you survive the acute fever and coma of a severe infection, your body hits a reset button.
The pathogen is cleared, and you are completely cured with no lingering effects. That's the myth.
Right, and the scientific fact, proven definitively by the MIND study, is what?
The fact is that certain severe infections, specifically cerebral malaria and severe malarial anemia, act as chronic neurodevelopmental risk factors.
Yeah, it's not a reset button.
No. The disease itself may be completely eradicated from the child's bloodstream, but the biological and structural changes inflicted on the vascular system and the brain echo for decades. They permanently alter cognitive development and academic potential.
This brings us to the real-world clinical and lifestyle implications. Global health policies right now focus massively on post-discharge chemoprevention.
Preventing the next infection.
Right. The primary strategy is to give children a course of antimalarials after they leave the hospital to ensure they don't catch malaria again while their immune systems are vulnerable.
Which is obviously crucial.
It is, but this study proves that it's fundamentally incomplete.
It's entirely incomplete if our ultimate goal is returning a child to their full potential. If we know, based on this peer-reviewed data, that a child surviving cerebral malaria has a highly elevated probability of facing sequential processing and mathematical deficits in school, well, educational and cognitive rehabilitation must be integrated into standard post-malaria care.
It has to be part of the prescription.
We can't simply discharge them with a pill and assume the job is done. Studies have already demonstrated that computerized cognitive rehabilitation training, these are programs designed to strengthen working memory and processing speed through targeted exercises, they can significantly improve short-term cognitive outcomes in children recovering from severe malaria.
It's basically physical therapy for the brain.
Exactly. By leveraging the brain's natural neuroplasticity, we can help them route around the damaged hardware.
But that requires resources, and a lot of them.
That's the bottleneck. The imperative now is for global health organizations to secure the funding and infrastructure to apply this cognitive support long-term.
It needs to be integrated directly into the educational systems and local clinics in malaria-endemic regions.
Otherwise, we're just saving lives, but ignoring the quality of those lives.
It's time to summarize today's deep dive. Severe childhood malaria, specifically the clinical phenotypes of cerebral malaria and severe malarial anemia, leaves a permanent biological imprint on the vascular system and the brain.
The result is diminished overall cognitive ability and math achievement up to 15 years after the acute illness has passed.
Right, and is driven by endothelial damage and blood-brain barrier dysfunction during that initial fever.
The actionable insight here applies to global health policy and local medical practice alike.
And what's that takeaway?
Measuring success strictly by lives saved is an incomplete metric. True recovery requires a massive paradigm shift. We must invest in post-illness cognitive and educational support to protect a patient's long-term quality of life and their fundamental academic potential.
Next time on the deep dive, we'll be examining the latest peer-reviewed data on how microplastics are crossing that very same blood-brain barrier we discussed today.
That is going to be a fascinating one.
And we'll cover what that infiltration means for the future of neurodegenerative diseases.
I want to leave you with a final concept to mull over. We spent today talking about a specific parasite in endemic regions.
Right.
But if a single mosquito bite causing an acute but temporary fever can invisibly alter a child's academic trajectory and brain hardware a full decade later, how many other temporary childhood illnesses, the severe flus, the high fevers we usually just brush off, are secretly shaping the cognitive landscape and the ultimate potential of our entire global population?
We tend to think of recovery as a simple return to perfect health. But as that 9-year-old girl from 1948 shows us, and as modern longitudinal data proves, sometimes the most profound impacts of a disease are the ones we simply don't have the tools to see until years after the fever breaks. Thank you for joining us on this deep dive.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.